Hi everyone, my name is Konstancja Urbaniak. I'm a postdoctoral fellow in Dr. Sergio Branchemore's group at City of Hope. Today, I would like to tell you the power of Bayesian network and our customized genomic software in diverse large-scale biological datasets. So let's first dive into what Bayesian networks are. These are a powerful probabilistic tool for modeling complex systems and making these predictions in a completely data-driven manner. Why would that matter in biology? Well, most biological systems, such as protein interactions, gene influence, this relationship is nonlinear. Therefore, it requires nonlinear approach to analyze it. The aim of this poster is to present the potential of the probabilistic Bayesian network in different cell-level datasets that can potentially lead to biological hypotheses. And the applications of Bayesian networks in research has been proven in multiple scenarios, including gene regulatory networks, system biology, image processing, disease progression, as well as semantic search. Bayesian networks all based on Bayes theorem, that is joint probability between different variables. And we can see different relationships between the variables, with the first one being a typical V structure. V structure means that this relationship is non-flipped in the directed acyclic graph that we're dealing with in our case, and in which the two variables A and C directly influence B, and they are dependent given B, while in the other scenarios, these graphs are equivalent and variable A and C are independent given B, similar here. And in the middle part, we have variable A and C are independent given B. But in this case, that looks more like a switch. When we see that A is on, let's say it's a light, the light is on, C is light off. If we observe the light is off, we know what's happening here. We know also what is happening here. So with the understanding of these Bayesian network graphs, and more specifically Gregory Gogoshin with Andrei Rodin, and they devised this genomic software that you can scan here and dive more into it. This Bayesian network model selection in genomic softwares addresses overfitting and underfitting. We want to create a balance. So the edge strength would be obtained that are obtained there are on the universal and bounded scale, which is the great advantage because then the Bayesian networks from different data sets, even different sizes, can be compared directly. And all is possible thanks to the novel addition to our software, which is the minimum uncertainty criterion. That criterion would allow for direct comparison across different data sets and different feature sets. And that is the part here. While the conditional mutual information is the basis for the Bayesian network, the conditional mutual information is derived from probability with the twist here that when we look at the conditional mutual information, there is computationally it's more efficient when we have subtraction rather than multiplication. And you will very soon see this suit for the genomics, and the downstream analysis also would be are already putting here. So soon would be available to the public as a suit for our genomics that you can see over there. So what can we do? One of the things that you can utilize Bayesian network is by just generating them. And we can do that on this very large scale when we put 2,000 variables. And then we can have a Markov blanket in order to select the variable of interest and see the parents of that variable and the children of that variable and the parents of the children. And that's how we can establish a nice Markov blanket and more concentrated view on the most important to us variables. Next, we can also, if the network is sufficiently small, we can generate it all and it would be visible all as it is. So there is this difference that one can have depending if we're dealing with thousands of variables where the picture maybe is less clear and requires the zoom in system, which is possible with the Markov blanket, or if we're looking at the very general network. And we can, with our Bayesian networks, we can very efficiently generate very multi-scale Bayesian networks from different omics data sets. We can do it on FACS, we can do it on RNA-seq, on spatial RNA-seq, bulk RNA-seq, of course. We can do it on the molecular simulations. So there is a variety of different biological data sets that can be put into our genomic system software, and very efficiently we can generate these Bayesian networks. What we can do with this Bayesian network? So there are multiple ways we can pursue with the analysis depending on the goal. One way would be to understand the typical causal interference. So if we affect each individual gene of our interest, let's say, we can predict what is going to happen to other genes in the network, and get a map of the affected genes and effector genes, and see what this correlation means, if they are upregulated, downregulated. We can do the interference also on the graphical level, on the gene level here, where we can, let's say, knock down 2 genes at once. We can knock out one, overexpress the other one. We can overexpress 2 genes at the same time, more, maybe 1, and that is the beauty of this causal interference, and see how the system would propagate the signal. We can, of course, do belief propagation as well. Or another way that is very commonly used Bayesian network for is feature selection. Feature selection is it's important for most of the large-scale data sets where we want to know what is happening in different settings. And in this one, as an example, we can see that the different features were emerging based on the group that they were assigned for. So if you'd like to know more, please have a look at these two references that are related to our custom Bayesian network, and more in depth into the math of how these networks were created, as well as if you'd like to know and experiment a bit with the Bayesian network on your system, you can do it while scanning this code, or this should be also available here. And I would like to express my sincere gratitude to everyone that was involved in the project, as well as the sponsors and also my supervisor. Thank you so much.